Good day, class. So for today's topic, we will be discussing about the general considerations and the overall introduction to what parasitology is. So let's get started. End of this lecture topic, the students will be able to know the different terminologies used for parasitology. You will also know the biological relationships in between the host. So later on, we will be explaining what a host is, and also we'll be defining what a parasite will be. The different types of exposure and infection. So how do you get or how is the disease transmitted from parasite to humans or to its um, host? Also, what are the sources of infection? The different modes of transmission, the life cycle of the parasite. But for this time class, I will only be teaching you the general life cycle of the parasite. Later on, when we discuss one by one the different types of parasites, then you will get to know the different life cycle of each parasite. And lastly, what are the different treatments involved in order to help in curing these parasitic infections? First, we need to discuss or describe or know the definition of parasitology. So, in the textbook, it says there that it's an area of biology concerned with the phenomenon of dependence of one living organism to another. It can either harm the other organism, it doesn't have any effect, or they both benefit with one another. So, that's the textbook definition of parasitology. But if you want to remember its definition by heart and so easily, you can simply look at the words parasitology. Parasitology, so you get the word parasite or parasite, and logy is a term that means study, so that's study of parasites, so parasitology. There are actually two areas of parasitology. We have medical parasitology and also tropical medicine. For medical parasitology class, it is concerned primarily with the animal parasites of humans and their medical significance, as well as their importance in the human communities. While for tropical medicine is a branch of medicine that deals with tropical diseases and other special medical problems of the tropical regions or from the tropical countries, right? So, under tropical medicine, they have something that they call tropical disease, and this tropical disease class is defined as an illness which is either indigenous or endemic to that tropical area, that specific tropical area, but may also occur in sporadic or endemic proportions in areas that are not even tropical. So it's very rare for it to happen that it can infect any tropical disease on areas that are not tropical, but it still is possible and can happen. Many tropical diseases are parasitic diseases in nature. As we all know, class, organisms may develop unique relationships with one another. That's due to our habitual or long associations with one another. And that is very evident even among us humans because we need to be out there in the community in order for us to survive. Remember, class, these relationships between organisms or between humans are very, very important. That's because for our survival. So for the biological relationships that we have, class, there are actually three types of symbiotic relationships. That's commensalism, mutualism, and even parasitism. But before we proceed to the next slide, because I will be showing you diagrams on what really is the difference between the three types of symbiotic relationships, what is the meaning of symbiosis? Okay, what do you think is the meaning of symbiosis? It's the living together of unlike or different organisms, and it may also involve protection or other advantages to one or both partners. And the different symbiosis class, it consists of all these other terms that we will be defining later on. So please, symbiosis is the living together of different organisms. Okay? Okay, let's start first with this first type of symbiosis, and it's called mutualism. In this diagram class, it's very easy for you to identify that both the species here, species A and B, benefit from one another. So this means that mutualism is a type of symbiosis in which two or more organisms, mutually benefit from each other. So they can really benefit from each other. Let's say, for example, the termites and the flagellates in their digestive system. So there are flagellates. You will know later on what flagellates are. These are type of protozoan parasites. So later on, for now, just know that these are some of the examples. This example here, it's a clownfish and a sea anemone. So both of the species can benefit from each other. So this slide here, class, are some of the examples of mutualism. So there are also bacteria that is found in our digestive system, and they also help us in the digestion and breaking down of food products and energy source so that it can be released to the body and be used by the body. So those are some of the good bacteria, the one that you found in the advertisements, right? So humans and the bacteria that is found in the gut. The bees and flowers, remember that pollination like that? So the bees can transfer one pollen to another, the clownfish and the anemone, the ants and aphids, lichen, woolly bat, and pitcher plant. So all of these are different examples for mutualism. The next type of symbiosis is what we call commensalism. So in the commensalism class, in the interaction that you can see here, species A or the other species benefits from the other species, but you remain the other species unaffected. So this means that commensalism is a symbiotic relationship in which two species can live together and one species that can benefit from the relationship between the two. So the other species can benefit, but the other remains unharmed or unaffected. So for example, the Entamoeba coli in the intestinal lumen, so it supplies nourishment and is protected from harm while it does not cause any damage to the tissues of the host. So, Miss, what's an Entamoeba coli? Later on, under the amoeba species, you will know what's an Entamoeba coli. But for now, if there are no problems with the host, if the host is not immunocompromised, meaning the host is healthy, then Entamoeba coli that is situated in the digestive tract or in the intestinal lumen of the host, then the host remains unharmed. Is that clear? So even if that's a parasite, if the host is healthy or not immunocompromised, then the host is unaffected. But um, the other species can benefit from it because it feeds there, right? So that's what we call commensalism. Let's show you what's an Entamoeba coli. Here's the picture of an Entamoeba coli in the stool sample of the patient or the host. So this is what an entamoeba coli looks like. Soon you will see this face to face under your microscope in our laboratory activities. But for now, just take a look at this structure. This is the nucleus of the entamoeba coli, and this is the entire structure of the organism. So if the patient again is healthy and non-immunocompromised, then it cannot infect or remain unaffected or unharmed. The host is remained unharmed, while this entamoeba coli is fed in the intestinal lumen of that host. So it can benefit. What type of symbiotic relationship does this have again? That's commensalism. And the last type of symbiotic relationship that we will be discussing is parasitism. As you can see here in the diagram class, the interaction is very clear that in parasitism, it's a symbiotic relationship where one organism, of course, that's the parasite, lives in or inside the body of the host or the other organism, and it depends on its survival on the host. And usually it's at the expense of the body of the host. So the species A or the parasite benefits from it, while the host or the body of the organism where that parasite lives is harmed. Okay? One very easy example of parasitism is a mosquito biting humans. Remember that female mosquitoes feed on human or even animal blood, so they inject or they bite us through our skin so that they can suck blood. So this mosquito benefits from us by biting our blood or sucking our blood, while we are harmed because we can either harbor different types of diseases like dengue, malaria, or also the itchiness that we feel right after. So that's another way of harming us, not just those other types of diseases. The simple itching is already at the expense of the host or us. So this is one example of parasitism. Another example of parasitism class is the infection of Entamoeba histolytica. I know everyone is familiar with amoebiasis, so this is the culprit for amoebic century. So it's nucleus here, and this is the organism. Also here, this is the organism with the nucleus here. This dark blue colors class are red blood cells. This will help us identify later on in your amoeba discussion. This will help you identify that this type of parasite is Entamoeba histolytica due to the presence of red blood cells inside. So they engulf red blood cells. And this Entamoeba histolytica class derives its nutrition from the human host and can cause amoebic dysentery. So this is another example of parasitism. These are the different parasites and their hosts that you can read some of this. By the way, this picture here, this is a hookworm. So later on, this will be discussed to you in nematodes. And this is what I really like about parasitology class. You can appreciate pictures and at the same time learn from them. And it's easier for us to like I'm not sure if you memorize, but it's easy for us to familiarize because there are pictures. And what's important is you understand the life cycle of this type of parasite. But for in this case, we'll have to, I'll just want to share to you that this is a hookworm and it's it attaches to the host's skin, so it can be humans, and it enters the bloodstream. Sorry. And this one is a fully engorged paralysis tick. This can also feed on the blood of mammals and can cause paralysis on the muscles where this tick enters. So these are the different types of parasites and their hosts that you can read some of this. Let's proceed to parasites. So we have been talking about what parasite is. So usually class parasites are described according to their habitat or mode of development. So let's discuss these different types of parasites one by one. Let's now define the difference between endoparasite and ectoparasite. An endoparasite class, this is a type of parasite that lives inside the body of a host. While an ectoparasite, from the word itself, it lives outside or on the surface of the body of the host. All right? So once you see presence of endoparasites class, you call that an infection. Presence of endoparasites or parasites living inside the body of the host, you call that infection. But for ectoparasites, the presence of that, once you find that in the surface of the body of the host, you call that infestation. Okay, so remember the difference. If it's an endoparasite inside the body, it's called infection. If it's outside the body or on the surface of the host, then you call that infestation. Right? Lice infestation of the hair, tick infestation on the fur of your dogs or your cats, infection, hookworm infection, because that is found in the bloodstream or in your gut or in your digestive system. So you call that infection. That's inside the body. Inside the body, endoparasite, you call that an infection. Outside of the body, it's infestation, and the parasite is called ectoparasite. Most parasites class are also called obligate parasites, and that they do not need a host at some stage of their life cycle to complete their development and to propagate their species. Obligate parasites such as tapeworms depend entirely upon their host for existence. So tapeworms, I will be showing you some pictures of tapeworms later. But an example of obligate parasites are tapeworms, and it means that you don't need a host at some stage of their life cycle. A facultative parasite may exist in a free living state or may become parasitic when the need arises. So facultative, you call that facultative. It can be free living, so it can live in lakes or in bodies of water, or it can become parasitic when there's a need. So you call that facultative parasites. And if the parasite, which establishes itself in a host where it does not ordinarily live, is called an accidental or incidental parasite. So it establishes itself in a host where it does not ordinarily live there. A permanent parasite class is the type of parasite that remains on or inside the body of the host for its entire life. Permanent, entire life. While for temporary parasite, it lives on the host's body only for a short period of time. So short period of time, temporary parasite remains on the body of the host for its entire life, permanent parasite. And there's also another type of parasite, which is a free living organism, and that passes through the digestive tract of the host without infecting the host. So you call that a spurious parasite. It does not infect the host, and it's a free living type of parasite. A free living type of parasite class means that it can survive in bodies of water or in the environment. So it cannot really affect or infect the host. Miss, you have been mentioning host, but we don't really understand what host is. Yes, class? Hosts in parasitology are the organisms where the parasite lives to thrive or to derive its nutrition. So, thus host class are classified into various types based on the role of the life cycle of the development of the parasite. So let's talk about the different types of hosts and define one by one. Okay, let's start first with the definitive or the primary host, or you can also call it the final host. This definitive host class is one in which the parasite remains or attains its sexual maturity. So, in the definitive host, that's sexual maturity. While for intermediate or secondary host class, this is where the parasite harbors its asexual or the larval stages of the parasite. So let's place tinea or tiniasis or a tapeworm infection as an example. So for the definitive or primary host of Taenia species class, it's the human as the definitive host, but for the intermediate host, it can either be pigs or cattle, depending on the type of tapeworm infecting the definitive host. So there's a bovine tapeworm, there's also a pig tapeworm. So we will be discussing that soon in cestodes. But for now, just know that intermediate or secondary hosts can be multiple. So there are probably first intermediate host, second intermediate host. This is where the asexual or the larval stages of the parasite develops. But in the definitive host, this is where it attains its sexual maturity. Okay, see now the difference between the definitive and the intermediate host. While for the paratenic host class, this is where the parasite does not evolve or develop any further to later stages. However, the parasite can remain alive and is able to infect another susceptible host. Thus, it's called a transport host, right? From the name itself, transport host, there are no life cycle developments that is happening here. It can only transport. It remains the parasite alive in its system, but it does not affect this type of host, but can infect another susceptible host. That's paratenic or transport host. While for reservoir host class, reservoir hosts, they allow the parasite's life cycle to continue and become additional sources of human infection. So they can be set aside there. The parasite lives inside this type of a host. It can continue its life cycle and an additional source of infection. That's the reservoir host. Miss, what's your example for paratenic or transport host? A very good example of paratenic or transport host class is a Paragonimus metacercariae in a raw wild boar meat that can pass through the intestinal wall of humans and complete its development. In this case, the wild boar serves as a paratenic host and it transfers the infected stage to humans once the humans ingest the raw wild boar meat. But who would do that, right? Or maybe some people would really do that in case of survival. And then they can be infected with Paragonibus metacercariae. How about an example of a reservoir host, Miss? Remember that reservoir hosts, they allow the life cycle of the parasite to continue inside and they can be possible sources of infection. So an example of this would be pigs are reservoirs of Balantidium coli. So later on, you will really know each one of them. But for now, these are just the different examples for reservoir hosts. So field rats, pigs for Balantidium coli, field rats for Paragonimus wistermani, and cats for Bruya malayi. Okay? Humans, however, are not always the final or definitive host. But as humans, we may be the most important host in the spread of the disease. And sometimes us humans can be accidental hosts of these parasites. Just accidental. It just so happens that you are infected with the parasite. So you call that accidental host. right? So those are different examples of the classification of under the classification of hosts. Again, what's a host class before I proceed? A host is the organism where the parasite lives, can attain its sexual maturity, can harbor its sexual or larval stages, or continue its life cycle development. Or the host can either be just transport, so the parasite can just live in the body of the host, but does not infect the host, but the type of host can infect another susceptible host and so on and so forth. All right, so please bear that in mind, the different definitions of the classifications of hosts. Aside from host class, we also have vectors. Vectors are responsible for transmitting the parasite from one person or from one organism to another. There are two types of vectors. There's a biologic or biological vector, and there's also a mechanical vector. A biologic or biological vector class, it transmits the parasite only after if the parasite has completed its development within the body of the host. Okay? It can only transmit once its sexual maturity has reached. A biologic vector, therefore, is an essential part of the parasite's life cycle. An example of this would be mosquitoes, Aedes mosquitoes. They transfer deadly diseases like malaria or even dengue mosquitoes from one susceptible host to another. So you call that biologic vector. And next would be mechanical or phoretic vector. Phoretic, Phoretic, that's mechanical or phoretic vector. This is the type of vector that transports only the parasite. An example would be what? Flies or cockroaches that feed on fecal matter, and they may carry enteric organisms and transfer these to our food. And this could be eaten by humans. So again, a mechanical vector, an example with this would be a fly or a cockroach. It can only transport or transfer the parasite, while the biologic vector, it waits for it to complete its sexual maturity before transferring it to another susceptible host. See now the difference? So an example of biologic vector is a mosquito, and mechanical vector is the fly. Okay, place that in mind, please. All right, recap. So a final host or a definitive host, it harbors the sexually mature parasite. How about the intermediate host? Immature or asexual or the larval stages of the parasite. How about the reservoir host? It harbors the same species of parasite as man and constitute as another source or a new source of infection to man or to another organism. So it's reservoir host. Usually it's animals. And vectors are arthropod that can transport the parasite to its host. So I hope you remember that. Let's describe exposure and infection. What do you think is the definition of exposure? It is the process of inoculating an infective agent. So you expose your host to an infective agent. And in the process of exposing your patient to this infective agent, you call that infection. And this connotes the establishment of the infective agent in the host, right? You establish already that there's an infection after exposure. And you call this type of infective agents as pathogens class. Remember pathogens in layman terms, we call them germs. Can either be parasites, bacteria, virus, prion, fungi, molds, right? So you call those Parasites that are harmful and can cause mechanical injury to the host as pathogens. There's also another term used in parasitology class. We call that a carrier. A carrier is a type of organism which harbors a particular pathogen without manifesting any signs and symptoms, meaning it's just a carrier. It carries this particular pathogen and tries to infect another susceptible host. But this type of carrier is not infected with it, it just carries it. All right? So you expose your susceptible host with a pathogen, probably through a carrier, and then the establishment of the exposure of the infective agent to the host is what we call infection. So is that clear? Is it not confusing? Okay, let's proceed. Let's discuss regarding incubation period. Miss, what's an incubation period? Incubation period class is the period between infection and the evidence of symptoms. So before you manifest the symptoms, there's still a window phase or an incubation period where the infective agent is fighting with the body's immune system and it takes time for it to like spread out. So you call that incubation period. There are actually two types of incubation period. One is clinical incubation period. So actually the definition that I have given a while ago was already for clinical incubation period. It is the time duration between the exposure to the pathogen and the manifestation or the appearance of symptoms of the disease. So you call that clinical incubation period. We also have a pre-patent or biologic incubation period. So either pre-patent or biologic incubation period. This is the period between infection or the acquisition of the parasite and the evidence or demonstration of the infection. Aside from that, we also have autoinfection and hyperinfection. What's the difference between the two? An autoinfection, from the name itself, is a type of infection which occurs due to larvae produced by the pathogen, which is already present in the body. In simpler terms, class, when an infected individual becomes his own direct source of infection, so you infect your own self, all right, that's autoinfection. Let's say, for example, in enterobiasis, so that's the kigwa in the lubot. Yes, that's enterobiasis. You call that enterobiasis, another nematode discussion that we will be doing soon. So the infection there is called autoinfection when you scratch your butt and then you accidentally like got food and eat before washing your hands. So that's auto-infection. There's hand-to-mouth transmission, auto-infection. You infected your own self. You are the source of your infection. So that's auto-infection. And for super infection class or hyperinfection, this is a type of repeated reinfection which occurs due to increased larvae migration. In simpler terms, it is it happens when the already infected individual is further infected with the same species, leading to massive infection with the parasite. So there's hyperinfection or superinfection. You are studied already with parasitic infection of the same species, right? So there's an increased number of larvae produced in the host's body. So that's the difference between autoinfection, you would become the source of your infection, while for hyperinfection or superinfection, you get too much or more parasites with the same species. For this slide class, these are the different classification of infections. If you can remember from the previous slide, we have autoinfection and then hyperinfection or superinfection, right? In here, there is also primary infection, From the word itself, primary, that means initial infection with the pathogen or organism to the host. For reinfection, there's subsequent infection, so subsequent infection by the same organism in a host, but this time it's after recovery. Compared to that of hyperinfection or superinfection class, that in hyperinfection, you get infected, reinfected with the same organism, and it increases the number of organism or parasites, parasitic load, right? But in reinfection, there is after recovery, and you get infected with the same organism. Superinfection, we have mentioned this, you get infected with the same organism in a host, but there is no recovery and there is increase in the parasitic load. Superinfection. For secondary infection, when a host whose resistance has been lowered, so unhealthy or immunocompromised by pre-existing infectious disease, then a new organism may set up an infection. What does this mean, miss? This can be nosocomial infection. Remember, nosocomial infection are hospital-acquired infection wherein the patient got a initial or primary infection um, from another organism, or let's say, for example, first bacterial infection. And then the secondary infection is already parasitic in nature. So you call that secondary infection. So can be nosocomial, you acquire the disease in the hospital. So those things really happen. And you call that secondary infection. There are actually quite a lot of various sources of infection class. And as you can see clearly from my PowerPoint presentation here, the photos here include a contaminated soil with trashes and garbage surrounding it because there is no proper sanitation, no cleanliness in that area. Probably it's unsanitary. So you expect very dirty land or soil. We also get different types of parasitic diseases from contaminated water. Not all of us have the luxury and access to clean and safe water class. So expect that some of the people get their water source from rivers and even lakes. Also, another source of infection is from raw or undercooked meat. It can either be a fish, pork, chicken, even chicken class, huh? And also beef. So it is always best to cook your meat before ingesting that. And in these cases here, class, you can see that in contaminated soils, especially when people don't have sanitary toilets or they use night soil or human excreta as a as a fertilizer, a fertilizer. So some eggs of certain nematodes such as Ascaris, Trichuris, Strongyloides, and even hookworm parasites can be found on the soil. So some kids or some children may run through this area here barefoot and they can therefore be infected with parasitic diseases. Remember, class, that you always need to be at all times follow proper hygiene and sanitary policies, Also for contaminated water, it can be contaminated with cysts of amoeba. I know some of you might be familiar of amoebiasis, so you can get it from contaminated water. Or it can sometimes be flagellates. So sooner or later, class, this will all be discussed to you. So for now, just get to know with the terms. So flagellates and amoeba are part of the protozoan parasites. For the cercaria, this is one life development of the trematodes. So we'll get to that soon. But for now, let's just know the sources of infection: contaminated soil, contaminated water, and also raw or undercooked fish, meat, or any type of meat. As what I've said in the previous slide, some sources of infection can be either from the food that we eat. So there are also delicacy in areas where paragonomyosis is endemic. And the intermediate host for this type of parasite class are crabs. So you can see from the diagram here, crabs. So if you fail to cook crabs properly, then probably there is a higher chance for you to get paragonomyosis. There are also raw bulastra snails. So this is what we call a bulastra snail. This one is associated with Echinostoma malayanum infections. So this is raw bulastra snails, raw crabs, and some vectors of malaria and other filarial parasites are mosquitoes, flies, sandflies. So these are the different vectors that could transport the carriers of the pathogenic parasites. All right, so some arthropods class are carriers of parasitic infections. And in this picture here, this is a triatoma bug, which is a carrier of Trypanosoma cruzi causing Chagas disease. This one at the center is a sandfly, and sandflies class are considered natural vectors of all types of leishmaniasis. In this picture here, it's a cat and a house rat. So for cats class, there are direct sources of Toxoplasma infection, while the house rats, they can transmit, they can infect you with Hymenolipis nana. Other sources of infection include asymptomatic carriers of Entamoeba histolytica, those workers that are food handlers in food establishments. That's the reason why, class, if you are working in a food establishment, there are certain medical checkups and physical examinations that are done to you just to make sure that you don't have any existing asymptomatic carrying diseases. Asymptomatic means you do not show any signs and symptoms of having this certain disease, so that's why you're called asymptomatic. So this is just to make sure that your food handlers don't carry any parasitic diseases within them, and they can probably transmit that through your food when you try to ingest the food that you ordered in any of those restaurants. All right. So those are probably some sources of infection. And even class your home beddings, the other person might be invited to your house and this person has parasitic infection, let's say enterobiasis. In Bisaya term, you call that kigwa. That's itching, it causes itchiness in your butt area. So what happens is that the eggs of the kigwa or Enterobius vermicularis from that other person who sits in your sofa or who sits in your beddings, it can probably transfer from their clothing to your beddings and then you can infect yourselves. So those are other ways of or other sources of infection or ways that you can get the infection. So clothing, beddings, other person. So those are some sources of infection for parasitic diseases. Okay, types of disease transmission. As we all know, class, since the most common source of infection is through contaminated food and water, then the most likely portal of entry of these parasitic infections would be through our mouths. Very good. So through the mouth. Okay, so we try to ingest these pathogens into our system. And we know that majority of infections among cestodes, trematodes, and even intestinal protozoans are foodborne. So we can get it through our food. Usually, examples of foodborne pathogens are Taenia saginata, so that's a beef tapeworm. Taenia solium, that's pork tapeworm. Dipelobotrium latum, that's a fish tapeworm. So those are, you can get that from raw or undercooked meat of those types of tapeworms. We can also get Entamoeba histolytica, so that's amoebiasis, and also giardiasis, Giardia lamblia, from contaminated drinking water. So our mouths are the most likely portal of entry for these types of the parasites. Even Clonorchis, Opistorchis class, this can be ingested through eating raw or undercooked freshwater fish, which contains the larval stages of this parasite. Aside by food, which enters through the mouth, it can also be through direct contact, or through insect bites. So that's skin transmission for direct contact and insect bites. That's skin transmission. An example for skin transmission would be hookworm infections, if you can remember. They can penetrate our skin from the soil if we walk barefooted. And also, aside from that, if we are in freshwater lakes, so there's also schistosoma, that's a type of trematode, which enters our skin, and that comes from the water. So we have to always be cautious wherever we go. So we're already done with by food, through direct contact. So this insect a while ago that I've said class, this is through vectors. Remember what are the purpose of vectors? They transport your carriers, which contains the pathogen. So these insects include like mosquitoes or other types of arthropods that would transmit the pathogen, and they might bite us. They can transmit malarial parasites into our system. So this is also another way of disease transmission. Also, class by air. If you can remember, I discussed earlier regarding the eggs of Enterobius vermicularis that could transfer from the clothing of the infected person to your beddings if they try to sleep in your area. Sometimes, class, if the load or the parasitic load is too much, you can inhale the eggs and get infected with Enterobius vermicularis. I know I've already said this a million times, but this Enterobius vermicularis is really very infectious, right? It's very infectious. This is the kigwa that I've said a while ago. You can also get some parasitic diseases through sexual intercourse. That's Trichomonas vaginalis. That's a sexually transmitted disease. So, Trichomonas vaginalis. Even Giardia lamblia can be transmitted through male to male sexual contact. So that's another parasitic disease that could transmit through direct contact. Another one is through indirect contact. Um, you might probably held onto something, and then that is very dirty, and then your hands were touching your utensils, so you're also be able to get those types of parasitic infection. So you can also get from that. There are also other ways in getting parasitic infections, class. There can also be congenital. So when a mother is pregnant, the unborn child might be infected with Toxoplasma gondii. That's an infection that is transmitted through cat litter boxes. So let's say there's a pregnant woman, and that pregnant woman owns a cat, and then that cat is infected with Toxoplasma gondii, and it so happened that this cat littered or poop in the litter box, and then the pregnant woman inhaled it or probably touched it, and then was able to ingest the type of parasite, there is a higher chance for that mother to infect the child, the unborn child, with Toxoplasma gondii infections. But later on, that will be discussed to you. Just for now, just a brief introduction regarding that. So that's also congenital way in transmitting the disease to the unborn child. There's also transmammary mode of transmission, disease transmission. Transmammary, that means there are certain parasites that could cause cross through the mother's milk. So there's a possibility that the child could ingest the parasite through the mother's milk. So that's transmammary. Okay, so we're already halfway through our discussion. So I hope you've understood the first 30 slides that I have discussed earlier. If you have any questions, save them for our face-to-face laboratory briefing. But for now, let's continue with nomenclature. So what's nomenclature, miss? So we all know that animal parasites are classified according to the International Code of Zoological Nomenclature. So that's International Code of Zoological Nomenclature. And each phylum class is subdivided into classes and which are further subdivided into orders, family, genera, and species. Right? So for the species class, I know that you're already familiar with this. Usually the scientific names of the species are Latinized. All right? You can see that there. And for now, just know that these are the different classifications of medically important parasites. Okay, so it's italicized also when printed. Remember, class, that in writing the species, the generic name or the first name, this area here, the first letter is capitalized, right? And Tamiba, that's the E there is capitalized, while the species name, it's, or the specific name, I mean, the specific name is in a small letter. So the generic name is capitalized and the specific name is small letter. So that's Entamoeba histolytica. If printed, that's italicized. So these are examples of the nomenclature. So make sure that in the practical examination, when you're asked to write the name of the parasite, this must be followed. The first letter of the generic name is capitalized, and the specific name is small letters, all of it. Let's now proceed to the general life cycle of parasites. So this means, class, that each parasite have their individual maturity or life cycle. This one is just a overview of the different life cycles. This is a general life cycle of the parasites. And usually, class, through adaptation of the parasite to their host and also the external environment, parasites have developed life cycles which may be simple or complicated. So most parasitic organisms attain sexual maturity where, again? In the primary or the definitive host. So they attain their sexual maturity in their definitive host, and some also spend their entire lives in the host or within the host's system or body. So while others are also exposed to the external environment, remember what I've said a while ago, there can be facultative parasites, right? What are facultative parasites again? These are free living type of parasites that could be infected once the need arises. All right, so that's a very good recap. So once they attain their sexual maturity, they can now bear eggs or spores, right? So the larval stages of the parasite may pass through different stages and can be transmitted either to another susceptible host or into the environment. And this time, this egg or spore, this stage, would be harbored, remember, in the intermediate or secondary host. This is where growth and development stage of the parasite happens. So this can either cause damage or harm to the host or remain it unaffected. Okay, remember that in the host, the intermediate host, right? And once this intermediate host is already ready to release it again into the environment, so the infective larvae, is able to infect the next primary host, and the cycle continues. So that is what happens in the life cycle of a parasite. The perpetuation of the parasite, a specific species of a parasite, depends upon its ability to ensure transmission from one host to the other. The parasite must therefore adapt to protect itself from the host defenses because we have our own immune defenses as well as certain external defenses like heat, right? Heating our food, disinfectants. So those are external ways of defenses that we can have aside from the immune system inside our body. So these parasites also tries to defend itself from the different defenses that our body and even the external sources are doing against them. So they must overcome any attrition in the species by producing numerous progeny. So that is what truly happens. It's also the same as how humans try to survive every day. So that's also how parasites survive. So much now for parasitology, its terms and definitions. Let's proceed to epidemiologic measures. So let's first define what epidemiology means. Epidemiology is the study of patterns, distribution, and occurrence of a certain type of disease. While incidence class, it is the number of new cases of that certain infection appearing in a population during a specific period of time. Another terminology that we should also be familiar with under epidemiologic measures is prevalence. Prevalence class, it is the number of individuals in a certain population estimated to be infected with a particular parasite species at a given time. While cumulative prevalence is the percentage of individuals in a population infected with at least one parasite. So these are the difference between prevalence and cumulative prevalence. Please take note of that. By the way, class, both of this is expressed in percentage, but for prevalence, it's the percentage of individuals in a population to be infected with a particular parasite. For cumulative prevalence, at least one parasite. That type of population is infected with at least one parasite, cumulative. Another term that we need to define is intensity of infection. This one refers to the number of worms per infected person. It can be measured either directly or indirectly and is usually referred to as the worm burden. So that's the number of worms that is found in an infected person. Well, morbidity, as we all know, I think you're all familiar with this term. This is the clinical consequences of certain type of infections or diseases that can affect an individual's well-being. So we use morbidity for that. For the treatment class, I think all of us know that the most effective precautionary measure as well as treatment for parasitic infection is deworming. So this is making use of antihelmintic drugs in an individual, or it is also given through a public health program. So sometimes in schools, the barangay health center or even the municipal or city health centers would go to different public schools to provide deworming tablets to the kids so that if ever they have any worm burden, they can expel it out. All right. Next is cure rate. Cure rate is the number of previously positive subjects found to be egg negative on stool or even urine sample testing or examination. This is making use of standard procedures at a set time after there's a deworming program or after deworming has been done. All right. So those are previously positive subjects that becomes egg negative on stool examination. Well, for egg reduction rate class, this is the percentage fall of egg counts after deworming based on examination of a stool or urine sample using a standard procedure at a set time after the deworming treatment. So egg reduction rate. Really have to do egg count. Well, for cure rate, you just have to check if the parasitic eggs are no longer seen in the stool or urine sample. So see now the difference between cure rate and egg reduction rate. So how do they see or how do we use this term soon? All right. Another term for treatment is what we call selective treatment. This involves individual level deworming with selection for treatment based on a diagnosis of infection or an assessment of the intensity of the infection, or sometimes it's based on presumptive grounds. So this can be used as a strategy in whole population or in defined risk groups. Another term is targeted treatment. Targeted treatment class is a group level deworming where the risk group to be treated may either be with a prior diagnosis, may be defined by age, sex, or other social characteristics, irrespective of the person's infection status. So that's targeted treatment. While for universal treatment, that's already on a population level. That's a population level deworming in which the community is treated irrespective of the age, sex, infection status, or other social characteristics. So if selective treatment, that's individual level deworming. For targeted treatment, that's group level, risk group level deworming. While for universal treatment, that's the whole population level deworming. Another term used for treatment would be coverage. So it refers to the proportion of the target population reached by an intervention or treatment. It could be the percentage of school-age children treated during a treatment day. So again, a coverage is the proportion of the total population that you want to treat for a certain period of time. Another one is drug resistance. So this is genetically transmitted loss of susceptibility to a drug in a worm population that was supposedly previously sensitive to this appropriate therapeutic dose. But unfortunately, due to some genetic changes of this type of worm, it has um, developed resistance against this certain drug. So that's what we call drug resistance. As for efficacy, that's the effect of drug against an infective agent in ideal experimental conditions. So that's ideal. While for effectiveness is the measure of the effect of a drug against an infective agent in a particular environment with specific classifications or requirements for ecological, immunological, and even epidemiological determinants. So these are the difference between efficacy and effectiveness. Also, class, effectiveness is usually measured by means of qualitative and quantitative diagnostic tests, which detect eggs or larvae in feces or urine of the patient after an optimal time interval, which is variable for each parasite because they don't have the same life cycle, they don't have the same effect on certain drugs. That's why they have to be given a certain amount of time, right? So the cure rate and the egg reduction rate are indicators that are commonly used to measure the effectiveness or to the reduction of the prevalence and reduction in intensity of infection of the patient, respectively. Let's now proceed to the prevention and control. So these are just terms that I've placed here that you just need to like understand so that it will be easier for you in the next parts of the discussion. So for morbidity control class, it is the avoidance of illness caused by infections. It may be achieved by periodically deworming individuals or groups known to be at risk of morbidity. So we have to control morbidity. Next would be information, education, communication, or IEC. This is a health education strategy that aims to encourage people to adapt and maintain healthy life practices. Also, there's what we call environmental management. This is the planning, organization, performance, and monitoring of activities for the modification of environmental factors or their interaction with human beings, with a view to preventing or minimizing vector or intermediate host propagation and reducing contact between humans and the infective agent. So there is environmental management. I believe all of these terms and all of these sentences are very, very easy to understand. So I hope you can place that in your mind and in your heart to understand all of this. This is very easy. Also, one of the most important prevention and control is environmental sanitation. So it involves interventions to reduce environmental health risk, including the safe disposal and hygienic management of human and animal excreta, refuse, and wastewater. It also involves the control of vectors, intermediate host, and reservoir of disease. It also covers the provision of safe drinking water, which is very important, and the handling of food or for food safety. Housing also, that is very important, which must be adequate in terms of location, the quality of shelter, and indoor living conditions. The facilities for personal and domestic hygiene, as well as the safe and healthy working conditions. So it is not only the government that is responsible to do all this, but we are also calling the help of everyone in our nation or in the community. Environmental sanitation. And lastly, for sanitation, it's a provision of access to adequate facilities for the safe disposal of human excreta, usually combined with access to safe drinking water. All right, so let's define first eradication versus elimination. What do you think is the difference between eradication and elimination? When we say eradication class, it is defined as a permanent reduction to zero of the worldwide incidence of infection caused by a specific agent as a result of deliberate efforts. Once this is achieved, continued measures are no longer needed. So it's completely eradicated. On the other hand, disease elimination is a reduction to zero of the incidence of a specified disease in a defined geographic area as a result of deliberate efforts. Continued deliberation, intervention, or surveillance measures are still required. So even if there's already reduction to zero of this particular disease in a certain geographic region, but still, there should be surveillance, management, and intervention done in order for it to be completely eradicated. But for now, an example of eradicated disease is smallpox and polio. But for elimination, an example of this is measles. Before I end my lecture discussion, do you have any questions, clarifications regarding Introduction to Medical Parasitology? If so, I have placed here the references. It's a Philippine textbook of medical parasitology by Belizario and also the clinical parasitology by Zeibig. If ever you have any clarifications, confusions, you may scan through these books. They are my references. Thank you so much for listening. I hope you've learned something new from me today. If ever you have any concerns, you can email me or reach me through these platforms. So, till our next discussion regarding parasitology. Thank you.